Studied about how different organisms get their food in your previous classes. The process of obtaining the food necessary for the health and growth is called nutrition. There are autotrophic and heterotrophic modes of nutrition. Now, let us study about autotrophic nutrition. These organisms, means autotrophs, synthesize food itself by using light energy and carbon dioxide. They are capable of producing complex compounds like carbohydrates, proteins, and lipids. Example for these autotrophs are generally green plants. The autotrophs are the green plants synthesize the food materials like carbohydrates by the process of photosynthesis. This process, that is photosynthesis, is known as universal food provider. By the photosynthesis, plants are getting their food materials. Many heterotrophs, the majority of the heterotrophs depend on their food for the plants. Hence, the universal food provider, that is photosynthesis. Production of carbohydrates by green plants in the presence of sunlight by utilizing water and carbon dioxide is called photosynthesis. This is the definition of the photosynthesis. So, production of carbohydrates by green plants means the plants with chlorophyll are only green plants in the presence of sunlight by utilizing water and carbon dioxide is called photosynthesis. Then what are the required? So already we have studied the definition. In that we came across these requirements. The first one is carbon dioxide. This is obtained from the atmosphere. The water is absorbed from the soil by the root system. Chlorophyll, which is there in the green leaves. Sunlight from the sun. This is the equational form of the photosynthesis process. The reactants are carbon dioxide, water. The products are glucose, that is C6H12O6, and water and oxygen. This process takes place in the presence of light and chlorophyll. It is known that the plants synthesize carbohydrates, the smaller and simpler ones, and from them more complex ones like starch and cellulose. Animals are not capable of synthesizing food and they have to depend on plants for the same. This is the transverse section of the leaf. The leaf is surrounded by an upper epidermis and lower epidermis. These both epidermis are covered by a thick cuticle. It is present above the epidermis and above the lower epidermis. On the lower epidermis, there are some pores or openings which are called stomata. These stomata are surrounded by two guard cells. The stomata are meant for gaseous exchange and transpiration. In the place of midvein, there are vascular tissues. Those are surrounded by a circle of cells that is called bundle sheath. Inside the bundle sheath, there are vascular tissues called xylem and phloem. Xylem conducts the water and phloem conducts the food material. The tissue which is present between the upper and lower epidermis is called mesophyll. The mesophyll is of two types. One is palisade parenchyma and another is spongy parenchyma. The mesophyll which is present below the upper epidermis is columnar or pillar-like by leaving very less intercellular spaces. This mesophyll is called palisade parenchyma. Then above the lower epidermis, the mesophyll cells are irregular in shape and arranged very loosely by leaving many intercellular spaces. This tissue looks like a sponge. Hence it is called as spongy parenchyma. Here the chloroplasts are seen only in the leaf. In the anatomy of the leaf, the chloroplasts are observed in palisade parenchyma and spongy parenchyma. So this palisade and spongy parenchyma cells are with many chloroplasts inside each cells. Now let us see the internal structure of chloroplast. It is the site where the chlorophyll is present. The chloroplast is discoid in shape and it is surrounded by two membranes, outer membrane and inner membrane. The space between these two membranes is called as periplastidial space. Inside the chloroplast, there is a granular substance called as stroma. In the stroma, there are flattened sac-like structures present. These sacs are called as thylakoids. These thylakoids are arranged one above the another. This structure is called as stacked thylakoids or piled up thylakoids. This, this piled up thylakoids or stacked thylakoids is known as granum or grana thylakoid. There are many granums or grana thylakoids in the chloroplast. All are interconnected with each other by a thin lamellae. The lamellae which are interconnecting the grana are called as stroma lamella. Stroma lamella. This is the structure of the chloroplast, and the chlorophyll, that is the light harvesting pigment. The chlorophyll is present in the granum or grana thylakoid. The pigments which are essential for photosynthesis, that is chlorophyll, are present in the granum. Photosynthesis takes place in two stages. One is light phase and another is dark phase. Light phase takes place in granum and the dark phase takes place in stroma. During the photosynthesis, there are several events takes place in the chloroplast. They are: first one, conversion of light energy to chemical energy. Light energy is trapped by chlorophyll molecule present in the chloroplast, then that is converted into chemical energy. Second, splitting of water molecule. The water molecule split into hydrogen ion plus hydroxyl ion in the presence of light. And third, the reduction of carbon dioxide to carbohydrates. Here, the carbon dioxide that is absorbed from the atmosphere is converted into carbohydrates. So, the carbohydrates are the products of the photosynthesis. So, here in these three, conversion of light energy to chemical energy and splitting of water molecule, these two changes takes place in the light phase of the photosynthesis. The reduction of carbon dioxide to carbohydrates takes place in the dark phase of the photosynthesis. Photosynthesis is completed in two phases. One is light phase and another is dark phase. Light phase is also called as light dependent reaction or photochemical reaction. Dark phase. It is also called as light independent reaction and biosynthetic phase. Then light dependent reactions. For this, light energy is required. This takes place in grana of chloroplast where the chlorophyll molecules are present. Assimilatory powers ATP and NADPH are produced in this reaction. Photolysis takes place. These are the four major events that takes place in light dependent reaction. The reactions that takes place in light dependent reactions will be studied under three steps. Step 1. In this, the chlorophyll is exposed to sunlight. It absorbs the energy present in photons and chlorophyll molecule is activated. What is photon? Photon is the unit of light energy which is present in the sunlight. Step 2. The absorbed energy used in splitting of water into hydrogen ion H plus and hydroxyl ion OH minus. This is called the photolysis. What is photolysis? It is splitting of water molecule in the presence of light energy. Photo means light, lysis means splitting. That is splitting of water molecule in the presence of light or light energy. This was discovered by Robert Hill. Hence, the reaction is called as Hill reaction. Step 3. Through a series of steps, the hydroxyl ion is converted into water and oxygen. This oxygen is released into the atmosphere. H plus ions undergo series of reactions. ATP and NADPH is formed at the end of the light reaction. ATP means adenosine triphosphate. NADPH means nicotinamide adenine dinucleotide phosphate. So, what are the end products of the light phase? One is oxygen is released. So, oxygen is one end product. And the assimilatory powers, that is energy rich compounds ATPs and NADPHs are formed in this light reaction or light. The next step is light independent reaction. It is independent of light. Means this reaction does not require the presence of light. This is also called dark reaction. But the term dark reaction or light independent reaction does not mean that they occur when it is dark at night. This light independent reaction or dark reaction takes place in stroma of the chloroplast. The NADPH and ATP which are produced in light reaction are utilized in this reaction. In this light independent reaction, RUBP, ribulose bisphosphate, is the primary acceptor of carbon dioxide because the carbon dioxide is reduced to carbohydrates or to glucose. Hence, carbon dioxide should be accepted by a substance, that is RUBP. By a series of steps, carbon dioxide convert to starch by utilizing the energy from NADPH and ATP. Now we will discuss about the experiments which are related with the photosynthesis. It is Hydrilla experiment. The aim of the experiment is oxygen is produced during the photosynthesis in the presence of light. The materials required for this experiment are a beaker, funnel, test tube, water, hydraulic wicks, matchsticks or incense sticks. Then the procedure: take a beaker and a funnel, take hydrilla plant twigs and keep it in the funnel. The twig edges should be inserted in the stem of the funnel. Then hydrilla plant along with the funnel inverted in the beaker. Then the beaker is filled with water. Then take a test tube and fill it with water and then invert it on the stem of the funnel. This is the setup of the apparatus for this experiment, and there is a precaution to take for this experiment. Ensure that the level of the water in the beaker is above the level of stem of the inverted funnel. So here it is the tip of the inverted funnel, and this is the level of the water in the beaker. The level of the water in the beaker always should be above the level of the tip or the edge of the stem of the funnel. This is the precaution. Then place this apparatus in the sun at least 2 to 3 hours. Then we have to observe. So what we have observed, a gas will be collected in the test tube. So here at the tip of this test tube, a gas is collected. So we are thinking that this is the oxygen. So how this oxygen is accumulated here? Because the hydrilla plant, by the process of photosynthesis, it is producing oxygen, and the oxygen is in the form of bubbles, reaches to the tip of the test tube and accumulated here. So we have to prove that the accumulated gas is oxygen. So for that there is a confirmation test. What is it? Test the gas in the test tube by inserting glowing matchstick or incense stick, which would burst into flame. If we keep a matchstick or incense stick which is exposed to the gas which is accumulated in the test tube, that burst into flames. This is the confirmation test for oxygen. By this we can prove that the oxygen is produced during the photosynthesis by hydrilla plant in the presence of light. A light screen experiment. The aim of the experiment is to prove the sunlight is necessary for photosynthesis. Materials required: potted plant, light screen or 2 strips of black paper and iodine solution. Procedure: take a potted plant with destarched leaves. The destarching of the leaves will be done by keeping the plant in the dark for nearly a week to remove the starch from the leaves. This is called destarching. Cover one of its leaves with black paper on which a design is cut. So here the S-type design is there. Fix the paper on the leaf in such a manner that light does not enter the dark part. Place this potted plant in sunlight. After a few hours of exposure to bright sunlight, test the leaf which covered by the black paper for the presence of starch. So detach the leaf and then remove this black paper strip and keep this leaf in a petri dish and add iodine solution. Here the part of the leaf which is exposed to sunlight only turns into blue. The remaining part which is covered by black paper does not change its color. This is due to here this part is not receiving sunlight. So the starch is not prepared because the photosynthesis does not occur without sunlight. So this experiment proves that sunlight is necessary to form starch in green plants by the process of photosynthesis. Mole's half-leaf experiment. A. To prove that the carbon dioxide is necessary for photosynthesis. Materials required for this experiment are a potted plant, preferably with a long and narrow leaves, a wide-mouthed transparent bottle, splitted cork, potassium hydroxide, and iodine solution. Take a potted plant, and this potted plant should be discharged. For discharging, we need to keep the plant in the dark for nearly a week to remove the starch from the leaves. After discharging, we have to take out the plant, and a wide mouthed bottle with a little amount of KOH, potassium hydroxide solution, is fixed with a leaf of the potted plant. Half of the leaf is outside the bottle and half of the leaf is inside the bottle. Then this apparatus is transferred under the sunlight and kept for 2 to 3 hours. Then the leaf with bottle is detached from the plant and then the bottle is removed. Then this removed leaf to be tested for starch with iodine solution. So keep that leaf in the petri dish and add iodine solution to the leaf and observe the changes of color. The part of the leaf which is outside the bottle turn into blue-black color, but the part which is inside the bottle does not show any change in color. So here this is the iodine test is to confirm the starch. Iodine turns starch into blue-black color. So this pot contains starch. So there is no change in color means there is no starch in the leaf. The reason is KOH present in the bottle absorbs carbon dioxide present in the bottle and the carbon dioxide is not available to the leaf for photosynthesis. Hence, starch is not produced in the part of the leaf which is inside the bottle and no change in the color of the leaf with iodine test. This confirms that without carbon dioxide, the leaves of the plants cannot synthesize starch by the process of photosynthesis. Hence, carbon dioxide is necessary for photosynthesis. Presence of starch in the leaf. The aim of the experiment is to prove that the starch is present in the leaf. Materials required for this experiment are a beaker, boiling tube, a tripod, a Bunsen burner, an asbestos gauze, a leaf, a methylated spirit, and water. Then procedure. Boil the leaf in the methylated spirit. Boil the leaf in the methylated spirit over a water bath. So this is the water bath. Water bath means the beaker along with water which is kept on the tripod and the water is boiled by the Bunsen burner. On this water bath, we have to boil the leaf in methylated spirit. So after some time, the leaf becomes pale white due to the removal of chlorophyll. So the leaf which is green in color will become pale white due to removal of chlorophyll from the leaf. Then take the leaf carefully from the test tube by using a brush. Then spread the leaf in the petri dish and add a few drops of tincture iodine solution on it. Again, observe the leaf. The leaf turns into blue black color. The iodine solution turns the leaf into a blue black in color. This is the confirmation test for the starch. The iodine changes the starch into blue color. Here the leaf turns into blue with the iodine solution, hence starch is present in the leaf. So this is the experiment to prove that the starch is present in the leaf.